Good morning from my side. I hope you can hear me. It is my pleasure to present RIANA Therapeutics today to you. What we do, we develop first-in-class inhibitors targeting transcription factor for cancer patients. And let me explain you how we do it. So RIANA is a university spin-off founded 2023. And technology I will present to you is coming from the lab of Professor Morigal and also my research still at the academic time. Our team is complemented by Christine Ruckenbauer, who is also in the audience, expert in technology transfer, who supports our scientific team. Our expertise in-house is mainly on molecular biology, STAT5 expertise, and also oncology and drug development. So expertise like chemistry that we do not have in-house, for example, medicinal chemistry, we are complementing from expertise from people like Peter Nussbaumer, currently at LDC, previously at Novartis, Anna Quattrapani, Novasign, and Anavo, Jürgen Moll, ex-Boehringer, expert in cancer development, and Oliver Sola and Michael Krebs support us with company foundation, finances, and business development. And the disease we're tackling is acute myeloid leukemia, still one of the deadliest blood cancers out there. You can see here that 5-year survival prognosis is still only 20%, and this is disease of elderly people, meaning it's very advanced, very fragile population, which is usually not eligible for stem cell transplantation or harsh chemotherapy. There are some targeted therapies emerging at the market with a market for such therapies of 1.2 billion euros. However, even if the patients respond initially in the beginning, usually they relapse very fast within first year. And once patient relapses, there's pretty much nothing else that can be done with this patient. And usually they succumb within like few months. So what we're trying to do is to target STAT5, a transcription factor, and it's validated target in oncology since already 20 years. And the challenge with this protein is that not only a potent oncogene like some of the market, but it's also a very essential protein. So the big challenge with this protein is how do we target its oncogenic properties without harming actually essential function, which are required for our blood formation, liver formation, and so on. And the unique thing that was discovered about STAT5 actually in our lab by Professor Morigal was that we managed to dissect these functions of STAT5 by different domain. So we postulate that STAT5 dimers mediated through dimerization domain are essential for STAT5 essential healthy function. But STAT5 oncogenic functions are mediated mostly through its oligomerization domain, so through a different domain of STAT5, which this actually allows us to build a strategy around targeting STAT5 that we can actually finally develop inhibitors for STAT5, which are so needed on the market. And that, of course, gives a huge opportunity because so far no one managed to do it. Key experiments that was done to validate this approach that it's really sufficient to block those oligomers to rescue cancer patients was done here in mice. And you can appreciate with the Kaplan-Meier plot that mice that have tons of oligomers really succumb very fast to multilineage leukemia and lymphoma. It is sufficient to delete this oligomerization domain or just point mutate it to completely rescue the mice and render them disease-free for over a year with low side effects. Which brings us to our hypothesis that if we selectively bring oligomers with small molecule inhibitors, we will kill cancer cells but without killing healthy cells, as they simply do not have those oligomers. And will provide the therapeutic window which is so needed for this fragile patient population. So also in terms of AML competition, there are quite few targeted therapies approaching now in the market. For example, kinase inhibitors or venetoclax BCL2 inhibitors, which provide great advantage to patients, but only until they relapse. And what's absolutely missing on the market is inhibitor targeting transcriptional level. And that's what we want to advance to such treatment as a combinational therapy, STAT5 inhibitors, where we can just fill the gap and hopefully inhibit the pathway once and for all and avoid this constant resistance development for the patients, because the cells would simply do not have any escape mechanism because like the whole pathway is blocked. And that, of course, will provide for patients more safe, more efficacious treatment, but also opportunity for us as we can develop first-in-class and hopefully eventually first-in-line therapy against STAT5. So what we've done so far, let me introduce you. As we started RIANA, we collaborated with Lead Discovery Center in Dortmund, experts in the early stage drug development, and did a quite huge wet lab high throughput screens, which after thorough validation gave us our first hits. We spent quite some time validating them with very thorough assays like cellular in our proprietary assay, functionals that we really kill in only cancer cells but not of course healthy cells, biophysical assay like binding to the recombinant protein by SPR, nano DSF. Also investigated chemistry to avoid any early red flags that would jeopardize our chemistry advantages, and of course early in vivo ADME. So right now we're in the middle of hit-to-lead optimization. We started a chemistry process, and already with our first cycle of chemistry, we saw quite encouraging results where our first generation of the optimized compounds have 10 times improved efficacy, but also improved selectivity, and also we already removed the first liability we saw in the chemical space. So this is quite encouraging results. And what are the next steps for our program? We plan to nominate 2 to 3 series for like further hit-to-lead stage, where I think we're already quite close to. Of course, continue medicinal chemistry to optimize our parameters and develop like a TPP product, and also introduce more of our product into standard of care therapies, because we really envisage this treatment as being part of standard of care treatment, combinational standard of care treatment for the patients. So we plan to reach proof of concept in the mouse model next year, and this would lead to eventually nominate lead compound in the beginning of 27. I also don't have time to show you, but we have quite a huge biomarker program running on the side to be able to really perfectly stratify patients once we're ready to start clinical trials. And I mentioned STAT5 for acute myeloid leukemia just because it's currently our focus as a very small, lean company. But STAT5 is a validated target in a number of other blood cancers, even solid cancers. And even more and more data now coming up about the role of STAT5 oligomers in autoimmune diseases. So once we developed a product for acute myeloid leukemia to the proof of concept, we plan to also expand in different indications. And moreover, we developed quite a unique assay setups that we use for screening for such difficult protein-protein interactions that can be also used for other targets with similar mechanism of action, like also protein-protein interactions, for example. So our clinical development strategy is fairly straightforward. At the moment, we're focusing on FLT3 positive AML patients, and once we have proof of concept there, we plan to expand in other blood cancers and eventually solid cancers. As I mentioned, our biomarker program also will allow us to stratify patients a bit more and maybe expand in different patient populations right from the beginning. This is our timeline. We managed to secure our pre-seed round of almost 2 million, mostly non-dilutively, which allowed us to start a company, do hit ID and validation, and even get quite advanced in hit-to-lead stage. We are currently focusing on the raise in our seed round with already 30% secured, which is in total 5 million and would get us to the drug candidate. And we're following quite straightforward drug discovery program, which afterwards will get us by the Series A to pre-IND and the clinical validation in human patients. So talking about our seed round, we now open to secure a seed round, and we already secured 30% of it. We also expect quite some non-dilutive grants coming in to leverage our funding. And right now we're looking for 3.5 million equity, which would give us to validate a drug candidate by Q2 2028, ready to start pre-IND studies. We are of course very open to talk about like smaller amount of funds to like which will get us to intermediate milestones. So just to summarize our USP about our STAT5 program, it is one validated target which can be applied to multiple indications. There is a significant market in AML, but there's of course can be also other diseases approached. We have a very clear path to clinical studies for this molecule, which will give high safety, strong efficacy for the patients. Our team is amazing and very complementary to each other. And yeah, if would be happy, please reach out to me if you would like to talk about our seed rounds or potential partnership. And thank you for your attention.